What's your favorite sea animal?
Narwhal. Oh, no, beluga whale. Definitely a beluga whale. I've actually swam with beluga whales, and it was the best day of my life. I'm currently a first-year PhD student at Scripps Institution of Oceanography, which is part of UC San Diego in California. I am a marine microbiologist by training, and I have studied microbes from the water column in the past, but I really find this project interesting because it's something that hasn't been looked at before. We're looking for piezophiles, which are pressure-adapted microbes from the subsea floor. I am collecting samples from every hole, one every 10 meters approximately, and I am going to take some of them and I'm going to put them in different types of incubations under different conditions in different medias, trying to select for very specific type of microbes. And then I'm going to take these incubations and I'm going to pressurize them. And no one's ever actually tested them at high pressure, so we're curious to see, what types of microbes are down there. Can they survive at these high pressures? And the ones that do, we're interested in looking at who they are, what they're doing, and just kind of the community as a whole. This is the hand pump, and it's used to pressurize pressure vessels, which we store samples in. The pressure vessels look like this. They weigh about 30 pounds each. They have this little pin right here that holds the lid in place. What happens when we pressurize is the lid gets pulled up because of the pressure, and then the pin stops moving, and that's how you know you have pressure in there. You have to fill it with water because it's water pressure, so it's actually a lot safer than using gas, because gases tend to explode. So I'm going to fill it with water. This is the very end of my process. So first I will prepare the samples in these little 5 milliliter glass vials, and then once they're ready to be pressurized, we bring them down here, we pop them in, fill it up with water. Watch out, it's gonna probably squirt water. Sometimes you gotta force it. Watch out. So that, because it squirted out water, we know that there's no air in the system, which is good. So we put our pin in. We have this nifty little holder so that it doesn't tip over in case the boat rocks. And this is our high pressure line that we're going to connect to the block. Now that that's on there really tight, I'm going to pressurize it. So when the system is open and I pump, water supposedly will come out, there you go, comes out of the outflow. And in order to pressurize it, you just close off the outflow and water will be pushed into the vessel. Now we know the pin won't move anymore, so we know that we're pressurized. And in order to remove the pressure vessel from the system, you have to close it off on here. And then we can remove this pin. And there you go. I just started my work, but I've done a lot so far, and I'm hoping to include a lot of what I'm doing here in my thesis.